Hello everyone, my name is Caitlin Murchison and I'm here to talk to you today about the regressive evolution in the teleost species Astyanax mexicanus, also commonly known as the blind cavefish. So to start off, regressive evolution is defined as the loss of useless characteristics over time. Cave animals or cave fish in specific are great examples to study regressive evolution just because they offer various advantages for studying evolutionary mechanisms and evolutionary change that we see in our present world today. For example, the teleost species Astyanax mexicanus include a surface dwelling and both a cave dwelling form as well. The numerous, and I mean numerous as in many different kinds, of Astyanax mexicanus, the blind cavefish forms, they evolve their recessive phenotypes independently than the rest of the surface-dwelling forms. The ancestral origin of the blind cavefish is very, very intricate, so I'm going to try to break it down as easy as I possibly can. The blind cavefish provide a very distinct model of evolution associated with populating a brand new habitat. This includes the sided surface fish, which populate the rivers of Mexico and southern Texas, while about 29 populations of the blind and eyeless, meaning they have no eyes, populate the Sierra del Abra and Sierra del Guatemala regions of present-day northeast Mexico. These are the blind cavefish that we are speaking of. The Sierra del Abra caves that harbor the blind cavefish arose during the mid-Cretaceous era, and this happened to expose an upper level of limestone that was not previously exposed. The limestone regions represent two major sedimentary environments, one an unexposed subsurface region and a younger reef. So the unexposed region's caves elevation is a physical barrier between the cave and surface populations. Due to the various caves having different elevations above sea level, some of the blind cavefish populations are more spatially isolated than others. In addition to the vertical isolation, an internal drainage system that is in place today also divides the populations from the northern and southern El Abra caves. An additional geological variable influencing the distribution of these species is the Central America Trans-Mexican Volcanic Belt, also known as the TMVB. It's just a site of intense volcanic activity, and it limits the gene flow between the populations in that area. Recent studies suggest that the blind cavefish populations have crossed this geological barrier around at least three times, and all three migrant populations have invaded the subterranean environment. The geological features associating cave populations influence morphological and physiological evolution of populations. The blind cavefish, as well as other cave animals, are useful in determining regressive and constructive traits because they are prime examples of regressive evolution. More specifically, the blind cavefish provides an excellent model for studying the developmental mechanisms of convergent evolution, while the genome of the Astyanax surface fish is a resource for comparative genetic studies of the trait differences between the surface and the cave dwelling species that we see today. In cave animals, the environmental cue leading to the eye and pigment regression is the absence of light. Eye and pigment regression is linked to the environmental conditions that have produced it. So just like we see in the surface fish where they have the light, obviously they're going to have eyes, they're going to have a lot more pigment, but the ones that are located in darkness, they don't really have the use for eyes. Therefore, we have regressive evolution. They don't need eyes, they can't see anyways, they have the absence of light, they lose their pigmentation. Let's learn more about this information from here. So, due to the Astyanax surface fish and the cavefish being infertile, the cavefish can be crossed for genetic complementation analysis. The cavefish are then compared with their surface fish, and we are able to identify the genes responsible for the trait loss in particular. Complementation indicates that about 12 gene loci are responsible for eye degradation in most cavefish populations, revealing a loss of function at 3 or 4 loci is enough to abolish the vision permanently. 3 candidate genes are found to be responsible for the eye quantitative trait loss, commonly referred to as the QTL analysis. The first candidate responsible is the alpha A crystalline gene, which is linked directly next to the eye QTL in linkage group 21. The second candidate gene, SHROOM2, is also located close to an eye QTL near the albinism gene and OCA2 gene linkage group 5. The SHROOM2 gene functions to aid in synthesizing and localizing melanin granules in the RPE. The final candidate gene is the ROM1, and it's also directly linked to the IQTL in linkage group 1. This gene encodes a structural protein required to maintain the flattened shape of the outer discs of rod photoreceptor cells. From all of this knowledge, researchers have concluded that since the candidate genes were located near the end of the developmental pathways, they are accountable for eye lens and retina differentiation, and that some genes at the termini of developmental pathways might be affected during eye regression in the blind cavefish. The loss of skin pigmentation is another primary regressive trait that we see within the blind cavefish species. In the surface-dwelling animals, pigmentation protects organisms from sunlight, aids in camouflage, sex recognition, mimicry, all of which are definitely completely irrelevant in the cave environment because none of that is necessary, none of that is even there. Sunlight is absent. So the Astyanax mexicanus has 3 pigment cells within the entire species. You have the black melanophores, the silver or red iridophores, and orange xanthophores. Over time, the cavefish developed a lighter skin shade just because there was fewer melanophores and a reduction of loss of melanin pigment in the environment that they were in. About 18 additional QTLs have been identified that affect the amount of melanophore-based pigmentation in cavefish. Albinism and brown gene mutations exist among the many QTL and predicted genes that express elements of melanophore and pigment reduction. The origin of the hypomorphic albinism phenotype can be found by identifying cavefish oculocutaneous albinism type 2, also known as OCA2 mutations. The OCA2 gene equips the body with instructions to properly assemble the P protein found in melanocytes. Melanocytes are specialized cells that produce the pigment melanin in our bodies. The variants in OCA2 gene disrupt the melanin production, which depletes skin and eye pigment and therefore affects vision. In the surface fish, 2-point mutations result in conserved amino acid substitution and a large deletion extending from intron 23 throughout the exon 24. And another example found was the Molino cavefish have 1 large deletion that encompass exon 21, which significantly condenses the OCA2 protein. In each example, the deletions are located in regions where the membrane spanning domains are predicted to drive the loss of function. Therefore, we see they're losing their function when there are mutations in these regions. While the eye loss and albinism have been studied extensively in the cave species, behavioral traits like sleep loss, alterations in foraging, reduction in social behaviors are currently being analyzed today to understand the genetic cause of behavioral evolution. So that is another thing to look into with these species that would be really interesting for future projects. Indirect selection could also be a probable cause for the cavefish regressive evolution of eye loss. Their eye loss could be lost due to their lack of need, while the constructive evolution of other beneficial traits was strengthened during that time. Also, genetic drift, it could be a possible explanation for eye loss. Studying distinctions in the regulatory genome reveals mechanisms linking gene function and evolutionary processes. In addition to eye regression, cavefish evolved larger sensory organs, for example, the olfactory epithelia, whose function is to specialize in detection of molecules associated with feeding and reproduction. Eye pigmentation loss in Astyanax is a selective advantage due to its ability to conserve energy and resources in a very low nutrient environment in their caves. Therefore, cavefish likely have two elements underlying the eye loss: selection to enhance survival in the darkness, and genetic drift by the random assembly of mutations in eye genes that are not counter-selected upon. The two morphotypes of A. mexicanus serve as models for evolutionary biologists to discern morphological and behavioral evolution in response to environmental modification. The selectivity and evolutionary mechanisms have provided researchers with an example to better understand evolution through comparative analyses and specify the specific characteristics that may have helped their predecessors colonize caves. Using this species as a model system has helped evolutionary biologists better understand the evolution of cave-related phenotypes. Well, that is all for now on regressive evolution of blind cavefish. Thank you so much for watching and have a great afternoon. Thank you.